So I'm about to introduce our last keynote of the day, and Dr. Savio Chan. He is our inaugural Gleason Neuroscience lecturer. Savio received his PhD in 2001 from the Chinese University of Hong Kong, and then he moved on to the United States to pursue a postdoctoral fellowship at Northwestern University. As a postdoctoral fellow, he received the honor of being the fellow of the Parkinson's Disease Foundation. He then moved on to a research track faculty appointment, and then he's been very successful, and he started his tenure track lab assistant professor position in 2012. He has been continuously NIH funded through an R01 since 2010, having the same R01 running for 9 years. For all of you that are sending grants out, it's an actual impressive accomplishment. He's also a great colleague. He never hesitates to send you an example of his funded grants, give you comments on your aims. He's a great mentor. He has developed some great resources for his lab members that he actually hasn't freely on his website. So I think it's important to emphasize those qualities in addition to great scientists that make somebody an all-around great colleague to be around. So welcome, Dr. Chan.
Can you hear me okay? Thank you, Lucy, for the very kind introduction. It's been a great day here. I enjoy the science very much. It's very impressive, and it's very different from what I do. But overall, I think the science at WSU is really outstanding. So I know everyone had a long day, so I will try to move my slides quickly. But if you have any questions, feel free to interrupt me at any point. Okay. So let's get started. So what we do in the lab is to study motor control from a cell and a circuit standpoint. Okay, so what I'm going to show you here, it's going to be a video showing how a robot actually moves a load onto a cart, a very simple task. But you can see that the robot actually failed, okay? So he actually bumped into a tray, and then his hand caught onto the cart, and the cart maybe fell, and then he also failed. So let's take a look. So you can see that he's trying to load whatever is in his hand onto the cart, but he failed. And then his hand got caught, and then he eventually fell on the floor. So you can see that this is kind of, I see it being really funny because you can imagine this is a very simple task for most of us, right? But imagine this is actually something that Parkinson's disease patients actually deal with on a regular basis. But overall, we don't fully know how to control our movement. It's actually quite complex, and that's what is captured in this video. So what do we mean by motor control? So I'm taking a very simple explanation or definition. It's basically how a brain or our nervous system regulate movement. Okay, so there are some known components to this. There's actually a diagram taken from a review from Silvia Arba and Rui Costa. So you can see that there's motor cortex up here, that's the basal ganglia, which my lab focuses a lot on. And down here is actually the brainstem and spinal cord. What's missing is actually the cerebellum. We know that cerebellum is actually very important for motor control. So, but what is the basal ganglia? So let's talk about that for a few seconds. The basal ganglia is actually evolutionary standpoint. From the evolutionary standpoint, it's actually very conserved, highly conserved. You can see similar or analogous structures, not only in mammals, but you can see that in birds, fish, and reptiles as well. We don't know a lot, except that we know enough that this circuit is actually important for spontaneous or purposeful movements. So in particular, in patients with Parkinson's disease, Huntington's, or dystonia, you can see actually disruption of the movement, and accordingly, you can see changes in the motor circuits or the circuits in the basal ganglia specifically. So I'm not going to go into the details of this map, but this is basically a clustering map taken from material carotene. So what basically it captures is the fact that basal ganglia is actually situated right here, away from everything else. So this is actually a clustering method describing the neuroscience field in general. You can see all the motor stuff are basically clustered together, except basal ganglia is actually away from it. On the other hand, you can see a bunch of diseases on this corner, including Huntington's disease and Parkinson's disease. But you can see that basal ganglia is actually nowhere close to this cluster. Right? There's something funny about the neuroscience field in general, and in particular with the basal ganglia. So a mission of the lab for the past couple of years has been set up to study the relationship between cell circuits and actions. So by better understanding how cells wire together in a natural state and how they're altered in the disease conditions by using disease models, hopefully we can actually create a framework to better not only understand diseases but also better treat diseases such as what I mentioned, Parkinson's, Huntington's, and dystonia. So let's look at the basal ganglia from a mouse. So here is the 3D reconstruction of the basal ganglia. And we are looking from the medial side of the brain. You can see that the big giant structure is called the striatum. It's something that we'll talk about a little. But the main focus of today's talk is actually this green structure, which is called the external globus pallidus, or the GPE. So if I'm looking at the structure itself, we can't quite appreciate the relative importance of the GPE yet. We can see that the striatum has to be really important just because of the sheer volume. And this is actually the input station of the entire circuit. But in terms of the GPE is relatively understudied, we think that this actually can be crucial to motor function and dysfunction. Okay, so here I'm highlighting one of the cells in the area. You can see that this single cell can actually form axonal emborization in a few different nuclei along the basal ganglia. So we can imagine that these cells can actually be important in controlling the output of the entire circuit. And we know that these cells are actually inhibitory. So potentially, these cells can be master or sending a master inhibitory signal, just stopping the transmission of signal across the entire circuit. But we don't quite know yet whether this is actually a common feature for all cells in the GPE. And I hope you get a sense from today's talk that we'll eventually tell you that this is not actually a common feature, but that's how we got started. Even back in the early 70s, from anatomical data as well as from electrophysiological data, people actually pointed out that there are some heterogeneity within the GPE. So here are some examples, and these are not early recording from the 70s, but perhaps from the 80s. These are actually recordings taken from a normal monkey sitting quietly in a chair. I think it was done back in the 80s. So you can see that there are three different units, three different neurons. They're kind of doing their own thing. They have different firing rates, and they have pauses here and there. And this cell obviously is firing a lot faster than the other two. Right, so again, it shows you there's some heterogeneity within the area. So on the other hand, this is actually a very consistent feature that people found not only in human patients, but also in animal models as well. So here, this is actually an MPTP-treated monkey, which is arguably the best model for Parkinson's disease. You can see that the firing pattern is actually quite different. Right, so all these three cells are actually doing something that are very close to each other. They are highly synchronous, and they're going through not only highly synchronous activity, they also have activity pattern that's very different from what we are seeing here. Right, you're seeing a spontaneous increase or decrease in activity here. Versus here's basically almost nonsense oscillatory burst and pause activity. And these examples are actually taken from a review from Hagai Bergman. Okay, so interestingly, These data kind of tell us that the GPE is clinically important. Especially we know that this activity kind of goes away with treatment that is known to be effective for Parkinson's disease, such as L-DOPA. When you give patients or animal models with L-DOPA, you can actually remove or reverse this kind of activity. Except that we know L-DOPA is actually very short-lived, right? So eventually we need, or patients actually need more effective treatment. So one of the, I would say, last resort for treating Parkinson's disease is actually implanting a stimulating electrode deep into the brain. In this case, it's actually the subthymic nucleus, which is an excitatory drive for the basal ganglia, including the GPE. So, but we can also imagine that this actually can be backfiring axons from the GPE as well. Right. So again, this is kind of a last resort. There's significant risks associated with the treatment itself. So hopefully, by understanding the disease better, we don't have to actually have patients go through this process. But just to give you a better sense of how effective DBS can be, so you can see this is right now a still image, but I'll turn it into video. So what it will show is actually this patient shaking his left arm. This is not voluntary. This is something that we call resting tremor. But once you turn on the deep brain stimulating electrode, you can actually see that the patient can actually control the more effective limb quite well, right? So how does it relate to the GPE? So again, we think the GPE is important from the clinical standpoint and also from the circuit standpoint. But what do we know about the GPE? So there's an early circuit model developed by Roger Alban that's taken from the late 80s. It's been 30 years, but we don't know a whole lot since. So basically what it's captured is here. The GPE is labeled as LGP. It's basically a black box. Right? It's basically a box with homogeneous population of neurons relating signal or information from the striatum to the STN, the subthymic nucleus. So if I am introducing too many acronyms or mentioning too many acronyms, feel free to stop me, okay? So this is quite boring. Can we do something about it? What can we add to the circuit, to the model? Finally, I want to just point out this really quick. The GPE is only proposed to receive one kind of input from the striatum, but we don't think that's the case. But I'm not going to talk about that very much today. So, but why do we think cell diversity is a good thing? Why is it meaningful, right? So think about in an orchestra, right? You can have a violin, that can be a basic unit to produce music. Why do you need so many other kind of instruments? Obviously, you can imagine if you add, let's say, viola, cello, or whatever, you can create an ensemble. You can make more interesting music, right? So we think our brain actually does something similar by adding different neuron types. We can actually perform more complex behavior. So perhaps I can give you a little bit of an example. Here, I mentioned earlier, striatum is the input station of the circuit, and there are actually two types of projection neurons. Okay, there are some cute acronyms for the projection neurons in the area. They either call SPN or MSN. SPN stands for striatal projection neuron or spine projection neurons. And MSN is kind of like the historical name. It stands for medium spinal neurons. Okay. So this is a more updated version of the same circuit model. So you can see that there are D1 neurons and D2 neurons, which is essentially something we call DSPNs or ISPNs, okay? And they produce two projection streams. One is called the direct pathway, and the other one is called the indirect pathway through the GP, eventually connected to the substantia nigra post-reticulata. So why is it important? Because now we know these neurons are actually differentially regulating motor control. Okay, so it's kind of an established but simplified view that the DSPNs are actually important for facilitating movement, while the ISPNs are important for suppressing movement. So you can imagine appropriate activation or activity of these neurons can actually crucial in controlling whether you move or not move, or how or the type of movement you produce. So we can imagine the challenges that we face when we study the GPE. We actually don't know what's in the area. At the beginning of our study, we didn't have any markers or any way to tell the cells apart. Okay. So, but then what's the consequence? So we don't know who is who. Obviously, you cannot just go back to the same cell and say like, oh, this is the same neuron that we have studied in yesterday, a week ago, a month ago, or years ago, right? And obviously, we cannot have across laboratory studies. We cannot have we cannot study the same neurons in different species, for example, and we also cannot link the individual neuron type to circuits and diseases. Okay. But how can we break down the GPE, or can the GPE be broken down? So different people have different ways, or have their own favorite ways to classify neurons, and it's all based on different attributes. So this is kind of an example of, say, how can we break down a tomato plant, right? It all depends on your favorite attributes that you want to pay attention to. Or you may say, I don't really care about cell type, I just want to know the output or the outcome. I just care about ketchup, right? So I don't think we're quite there yet. So we want to link cell type to the outcome, actually, or the behavior. So in terms of our metrics, we like to find molecular markers that we can kind of nail down individual cell types. For example, a protein marker, right? We actually study the intrinsic properties a lot because it's kind of a highly quantifiable way to tell cells apart from each other. We also look at the synaptic partners, both input and output. We also eventually want to map all the cells onto different behaviors, right? Or whether they relate to motor function, for example, and how different cell types are actually altered in disease conditions. Okay. So going back to the GPE, can that be broken down? So these are some of the early experiments I did when I was a postdoc. So these are not the higher throughput experiments, but it kind of gave us some clue potentially. So in this case, we have injected a dye into the striatum, and we have labeled cells into the GPE. You can see that the cells can actually send an axon back to the striatum without surprise, right? And it has some interesting electrophysiological features. But again, and you can imagine this very low throughput. And there's another interesting method, is single cell RT-PCR. We can actually harvest an individual neuron, and then we can actually tell cell apart. For example, we can tell GABAergic neuron apart from cholinergic neurons, right? But to be able to break down cells within this class, it becomes really challenging, and also becomes really, really slow. So luckily, a major breakthrough came about in 2010. The two papers back-to-back, one from John Rubenstein and the other one from Oscar Marin. They both had something similar. They said that the GPE is actually quite diverse. Neurons are actually coming from different developmental origins. And interestingly, they can be defined based on the expression of parvalbumin PV, which is a calcium-binding protein. We're only using it as a marker. Similarly, we're using MPAS1 as a marker. MPAS1 actually is a transcription factor. So some of you may know there are a lot of mouse lines that actually allow us to label or to manipulate cells that express PV, but there weren't anything available that allow us to identify or to manipulate MPAS1 neurons. So we decided to do something bold. So we made a mouse. So this is actually a back transgenic mouse that expresses Cre as well as T tomato as a reporter. As expected, the label cells in the cortex and hippocampus, so you can see all the dark dots. Those are actually tomato signal reporting the presence of cells that actually is expressing MPAS1. And importantly, if you blow up this little area, you can see the GPE here, where there are a lot of label cells as well. And interestingly, there are not a lot of stuff around here that has labeling of tomato or expression of Cre, so which is really neat. That will allow us to manipulate or identify cells quite specifically within the GPE. But how do we know the mouse actually worked? Right. So to do that, we have to make new antibodies, and we were able to do that. We succeeded. So for example, we knockout validated the antibody. We didn't see any labeling here, but you can see that the guinea pig and rabbit antibody label the same step, and accordingly, the antibody also label the same cells that were reported by the mice as well. Okay. So the first thing we want to do with our tool is to repeat what was reported before. Can we tell that PV neurons and MPAS1 neurons are different from each other? And that's kind of what we're showing here. We're showing sagittal mouse sections going from lateral to medial. These are low mag images, these are high mag images. I think these are a little too dim to look at, but let's draw your attention to here. You can see that magenta and green are non-overlapping, essentially. Okay, in this case, magenta is actually MPAS1. You can see it's actually a nuclear stain. That's why they look a little smaller. And versus PV cells are a little bigger. They're in general a little bigger, but also the labeling pattern is cytoplasmic. So what about the reverse or the inverse of the experiment, right? So here we are using PV antibody on our TD on our MPAS1 mouse, and again you can see that magenta and green are essentially non-overlapping. Only occasionally you can see one like this. Okay. So then you want to ask, okay, is the GPE very simple? Is it just containing two cell types? Is it dichotomous, right? And obviously, this biology, this is too good to be true. It turns out there are more than two cell types. So at the very least, we have PV cells, MPAS cells, and cells that are negative for both, okay? And from the literature, we also know that there are cholinergic neurons in the area. So this is roughly 5%, and this 50%, this 30%, so that left us maybe around 15% of the cells that are not molecularly defined yet. At least we don't have a unique marker for that population. Also, I want to point out that FOXP2 is actually a subset of MPAS1. Okay, so that information will become useful later on. Let's try to keep that in mind. So there's some quantification I'm not going to go through with you because it's just going to take up too much time. This kind of something that we're doing in progress. We're finding new markers for different cell types, and I'm not going to go through these images, but just to tell you that that's ongoing, and that's kind of the landscape that we're dealing with now. Okay, but again, mostly we're dealing with two classes of cells, so hopefully that idea will come through. So one way to look at different cell types is to actually look at the number, the density, as well as the distribution in the area. That's one way to quantify it. You can see that PV cells are clustered kind of rostrally in the more interior area. You can see that the area of the GP that's not occupied by the PV neurons. On the other hand, you can see MPAS1 neurons down here. They're quite evenly distributed within the GPE. Okay, and we think this is potentially a useful way to tell cells apart from each other. For example, the chat neurons, the cholinergic neurons, are actually displayed fairly caudally and ventrally, right? So, like I said earlier, that we actually like electrophysiological analysis because it's highly quantifiable and also it actually can tell us meaningful information. For example, this is a PV neuron. You can see that this a PV neuron in the slice prepared from a mouse. You can see that the activity is actually quite high relative to the MPAS1 neurons, right? The MPAS1 neurons have relatively low activity, and the activity pattern is relatively irregular. And it's being quantified here, okay? The rate for PV is high, MPAS is low, and then for variability, you can see that MPAS is significantly higher than PV. Okay, it's capturing what's being shown here. I'm not going to talk about those. But like I said earlier, we're looking into subtypes within PV and MPAS as well. So one useful way is to just grab all the different electrophysiological feature and throw it onto a computer algorithm and see what the computer tells us. And that's essentially what we did here. This kind of a hierarchical clustering analysis. It looks complicated, but basically it is just splitting the data in a binary way. Every time it is bifurcating. That's kind of what the dendrogram is illustrating. So if you look carefully, this and that branches actually split right around here. So these are all the AMPAS1 neurons and the related cell type. And these are all the PV neurons and the related cell type. You can see this is mostly brown and this is mostly pink. So pink is PV and then brown is AMPAS. Okay. So there's some subtle differences between subtypes, but overall we can categorize cells into PV and AMPAS. So early on, we have actually made an observation that if you record from a chronic 6-HD lesion mouse, which is a well-established rodent model of PD, we can actually see cells within the GPE with lower or decreased activity. Okay, so this is actually a histogram. We're plotting activity on the y-axis. We're plotting the fraction of cells on the y-axis and the activity level on the x. Okay, you can see that this is naive and this is the PD model. So a bunch of cells firing at a lower rate that you don't typically see in the naive mouse, right? But before transgenic mice become available for the identification of GP neurons, we don't know who is responsible for this, right? So, but now it's quite clear that from using the PV mice and the AMPAS1 mice, we can tell right away that the PV cells don't change in the disease model versus the AMPAS1 neurons have a lower firing rate. So we don't quite know yet what that means and what is underlying that, but you can tell just from the electrophysiological features and the alterations in the disease that these cells are actually different from each other. So there are a few other ways to quantify the data. Maybe I can just draw your attention to here. You can see that AMPAS1 negative neurons, which roughly correspond to PV positive neurons, don't change in the disease model, versus the AMPAS1 positive neurons or the PV negative neurons change the activity level in the chronic PD model. PD stands for Parkinson's disease here. So as I mentioned earlier, that the axonal projection pattern or the anatomy can actually be a useful feature to define cells. And here we're looking at the striatum, and we're looking at whether PV neurons or AMPAS1 neurons in the GP project to the striatum. And immediately you can see that there's not a whole lot in the striatum, and these are the blue stands for represent markers that label interneurons in the striatum. You can see that these axons actually targeting interneurons within the striatum. Versus AMPAS1, you can see a very high density of axons. Okay. So if you look at the STN, the subthalamic nucleus, you can actually see the complete opposite. You can see a lot of axons from the PV neurons, and there's high MAC images. You can see all the cell bodies are basically wrapped around by the PV neurons. Versus the AMPAS1 neurons, they don't actually go into the STN too much. They actually just skip over and went further downstream. So there's another way to look at the same anatomical features, except that we have also included a stain for VGAT, which is the vesicular GABA transporter. So we can actually tell the axons in the area actually produce scientific terminals, not just sending axon passing through the area. So you can see magenta is actually VGAT, right? So when magenta and green overlap, produce white, something like this, you can tell there's a putative release sites. And there are just a lot more axons and release sites produced by the PV neurons versus the AMPAS1 neurons in the STN. So this, like I said, a functional analysis or electrophysiological analysis, which again is highly quantitative, and we're looking at, we're monitoring neurotransmission here. So we're recording from an ST neuron, and we're looking at the release of GABA on to the ST neurons by the parvalbumin input or PV input. So these are our currents, and probably because of the type of electrode we are using, and we can tell from direction of the current as also from the sensitivity to GABAergic blockers. This is actually mediated by the release of GABA. And again, AMPAS1 neurons don't actually talk to the STN too much, right? Which is consistent with the anatomical data. And finally, it's being summarized here as a box plot. There are other ways to look at connectivity, and here we've done retrograde labeling. I'm not going to go through that too much. So what about the striatum? We mentioned earlier that the striatum actually received a lot of M+1 input. We actually think this is actually something we want to study rigorously, because the mouse is a new tool that allows us to study this. Like before, the availability of the M+1s is not something that anyone can study in the past. So these two images are similar to what I showed you here, but in the next slide, I'm going to summarize or quantify the data here, right? Again, you can see that M+1 just made a lot of axons into the striatum, right? As I mentioned earlier, the striatum is actually a major input station of the basal ganglia, right? So if M+1 is inhibiting the striatum, it can actually produce, it can actually be important for controlling motion or movement, right? So let's see how M+1 neuron talk to the projection neuron within the M+1. That's kind of a goal here. So similar to the image quantification, you can see that both DSPN and ISPN, these are the direct pathway neurons, and the indirect pathway neurons actually do receive input from the M+1 neurons, but not so much from the PV, right? So let's look at the ISPNs for a second. So M+1 is high, PV is low or non-existent, which is similar to the anatomical quantification, right? We can see that the connection to the ISPN overall is stronger compared to the DSPN. And here's the underlying data. The direction of the current is slightly different from, or is different from before, probably because of the electrode being used. This is to maximize our driving force, right? And for those of you who are familiar with this type of recording, you can see that the kinetics or the speed of these events are on the slower side. It's typically 2 to 5 times slower than what you expect to see. I'll come back and explain why they're so slow and the significance. So our lab actually study Parkinson's disease a lot. We want to understand how cells and circuits are actually altered in the disease condition. So these two recordings are similar to what I just showed you in the last slide, but you can see that in the chronic 6-HD lesion model of Parkinson's disease, the connections should become just much stronger for both cell types. And I mentioned that very early on that these cell types are actually important for facilitation of movement and suppression of movement, right? So if you have inappropriate inhibition of the activity of these cells, it's not hard to imagine that patients will have trouble moving or there will be hypokinetic symptoms. And that's how we're interpreting the data. Okay. So at the bottom, show you the way that we can quantify the data. And you can see that the DSPNs are on the left and the ISPNs are on the right. Essentially, there's a typically 2-fold increase in the response amplitude following the chronic 6-HD lesion treatment. And there's another way to quantify the data, but I'm not going to bore you with that. So, like I had mentioned, M+1 is GABAergic, right? These neurons should release GABA and inhibit the firing of activity of SPNs. So let's take a look at the data. So this what an SPN will look like. Typically, when you inject current, they produce action potentials. That's how they talk to each other or to downstream target areas. So here is a similar type of recording with the same current injection, except that here we're using light to activate the input of the GPE. And you can see that there's a suppression of spiking, which is not that surprising, right? And this is kind of reassuring. By adding GABAergic blocker to block GABA receptors, we can actually reverse the phenotype, right? So you can see them, the firing is restored when we added the GABAergic blockers. So this is all reassuring, except that when you look at the population data, you can see overall there's a stronger suppression of spiking in the lesion model, except that in the control condition or in the naive condition, the suppression of spiking is relatively weak. We were puzzled by this for a really long time until we went back and think, okay, why do we have such a strong projection. If you look at the anatomical data, there's a ton of axons in the area. How come the suppression of spiking is so weak, right? Something is not making sense until we make the connections between the kinetics of the events. We think the synapses are probably not on the cell body, and the input is likely not meant to be there to suppress somatic spiking. What do I mean by that? So we fill all the SPNs, all the spinal projection neurons, with a dye and image them on the spinning disk confocal while we record from them. And you can see magenta is actually coming from the Alexa dye, and all the green stuff is the MPAS1 axons coming from the GPE. If you look closely, there are a lot of white areas which I highlighted with arrowheads. And this is kind of an example taken from this little area. You can see this an axon of an SPN. You can see this very spiny, so which is again telling us this is truly a spiral neuron. And this is an axon making that's produced by an MPAS1 neuron from the GPE, and it's making two contacts onto this area. Okay, you can actually captured as a Z stack. It's a volume, so you can actually rotate the image a few different ways. And these are the two orthogonal views from the same structure. You can see that the white stuff produced, right? Again, it is essentially the overlapping of the green and the magenta. So you can see the same structure in three different orthogonal views, telling us that this is likely a truly putative contact, right? But you can always argue that this may not be real because it's a confocal analysis. So I will show you electron microscopy in a few seconds. So, but let's quantify this data a little bit. So you can see that most of the contacts are actually out at around 70 microns. Okay, and 99% of the contacts are actually on the dendrites, but not on the cell body. So now it becomes clear that they're likely not to be there to control somatic excitability of SPN. So there's the EM data, like I mentioned earlier, essentially just telling us that the same thing. You can easily find MPAS1 axons making contact with SPN dendrite. And if you're an expert in electron microscopy, you can actually tell easily that this is actually a very unique structure of dendrite, and you can tell this dendrite is actually coming from the spiral neuron. For example, if you look at the base here, there's actually a spine here. And if you move through the host, you can actually see there may be a spine coming over here as well. Okay, so here's how we can assess the function of MPAS1 on SPN spiking here. So instead of using current injection to drive somatic spiking, we're actually asking cortex to drive spiking. In this case, we're using electrical stimulation in the cortex, right? And then and that drives spiking in SPN. And you can see that these are all driven by the cortex. There's a failed spike. There's some kind of atopic spiking just happened to be there. The data overall is quantified as a raster plot. So what happened when we activate MPAS1 input? You can see that the spiking is heavily suppressed, right? You no longer see as many spikes, right? And also if you look at the raster, it's quite different from what you see here. And overall, there's a relationship, a negative relationship. It depends on the initial spiking probability, okay? So what do you mean by that? So initial spiking probability is, you can see as a measure of the strength of the cortical input, right? So whether the MPAS1 input can gauge the cortical signal actually depends on how strong the cortical signal is. That's essentially what it's plotting. So we can actually have a larger pool of data, and that's what we are showing here. You see a hotspot here. So when the cortex is really strong, the MPAS1 input cannot gate it. Okay. Versus here, when the cortex is really weak, the MPAS1 can just shut down the activity really effectively. Okay. There's some weirdo here. I'll get back to that in a bit. But otherwise, overall, the behavior is generally graded. And I'm not showing you the underlying data, but now we know that this is actually a very important feature that is maintained in the disease condition. So MPAS1 is always gating the cortical input. So remember, I showed you a couple slides ago, the M+1 input is actually strengthened in the chronic PD model, right? So it turns out it's there to give the strengthened cortical input. But potentially, because of the fact that the M+1 input is strengthened, it may be maladaptive. I'll get back to that in a few slides. So since the input is actually impinging onto the dendrites, we don't have electrical access to truly know what's happening. So currently we're using computational modeling to understand what's actually happening out there and what are the features that are critical for the interaction. It's not known yet, but it's something that we want to explore. So remember there are some weirdos in the last slide, right? Let me show you one more time. Remember these weirdos, right? They're not conforming to general behavior. What are those? So it turns out the M+1 axons actually produce a very interesting turning pattern in the striatum. So you can see there are a lot of holes in the striatum that don't actually get inputs from the M+1 neurons. So if you look at those holes carefully, it's actually quite consistent across the animals. Okay. And if you do a staining for mu-opioreceptors, you can actually see that all those holes actually reactive or immunopositive for mu-opioreceptors. So those are the subcompartments within the striatum that's called patches or stereosomes. So we know that 90% of the striatum is called the matrix. So that's important for motor control or motor function. Versus the stereosomes or the patches, they're actually more related to limbic function or associative function. So these images essentially just argue that M+1 input has to regulate motor function. We'll get to that soon. So with a collaboration with Dave Levinger, we have made recordings from the stereosome area versus the matrix area. Again, it kind of tells us the same data as the anatomical data. Essentially, there's a very strong biased input from the M+1 axons or neurons to the SPNs in the matrix area, but not so much in the stereosome. So, but how can we assess the function of M+1 neurons? We kept thinking they have to be important for controlling movement of animals. So one way to do that is to infect the M+1 neurons with a DREAD receptor virus. DREAD is actually an engineered receptor, so that is not actually responding to any ligand endogenous to the mouse, but the ligand can actually activate the receptor. It's called CNO. So when you administer CNO into the mouse, you can actually activate the receptor. And the drug receptor that we are delivering into the animals is actually called HM3DQ, which is an excitatory receptor. So by administering CNO into the mice, we are essentially driving the activity of M+1 neurons. But let's see what happened. Let's go through the data together. So this is a group of control mice. So you can see that CNO actually doesn't change the motor activity of mice. And on the y-axis, we're plotting motionless or freezing behavior, which has strong implication to Parkinson's disease. And a coronary vehicle also doesn't do anything. But if you look at the HM3DQ group, you can see the CNO increase freezing quite significantly, right? And that effect is not produced by vehicle. There's another way to plot the data, and we're plotting the freezing behavior in CNO versus baseline, okay? And each marker represents a single animal. You can see that the circles are all the controls, they're hovering along the no effect line, versus all the crosses are basically squished toward the y-axis, meaning that they all responded to CNO, except maybe this guy. So this is kind of nice, except that we can be missing stuff because this is a technique called chemogenetics. You can imagine once the drug is administered into the animal, it doesn't it doesn't go away for a couple of hours. So we cannot look at fine movement in this way. So we turn into optogenetics. So for those who are less familiar with optogenetics, basically we are putting light-sensitive channels or pumps into the neurons by using viruses, okay? And by shining light, we can actually turn things on and off quite specifically, both spatial temporally. So here we're looking at PV neurons, and let me walk you through the schematic because the rest of the figures are laid out in a very identical way. Okay, so we're dealing with PV neurons that's being labeled on top, and you can see the symbol or the little cue drawing here. The light is being delivered to the cell body of the PV neuron. And again, the downstream target is STN, like we have talked about a few times. So let's see what happened. So when we shine light, which is the blue box here, we can see the activity of the animals went up quite clearly, right? We're plotting the animal activity here, and we're plotting time over here. And if you do the same thing with PV neurons that only express YFP, which is a control, you can see that there's no change in activity level during the light stimulus, okay? So if you stimulate the terminals of the PV neurons in the STN, you can see something very similar to what we saw here. Okay, again, telling us that this is the circuit that is important. So we did that probably because of we want to be rigorous, and also we also know that PV neurons not only send axons to the STN, but also somewhere else. Okay, so what about this? Here we're expressing GT802, which is an inhibitory opsin. We put that into the STN. The reason to do that is to mimic the inhibition produced by the PV input. So when we shine light directly onto the STN, opening an anion channel, and we saw same thing, an increase in movement. Again, confirming that the stuff that we see here is truly through inhibition of STN neurons. Okay, so again, we're looking into a subtype within the GPE. Now we've found casein G4 being a potentially useful way to identify a subtype, and essentially it's showing us something similar. This is actually a subtype of PV neurons. And just there's a histological confirmation showing that the opsin is expressed in the GPE, and we have appropriate placement of the fiber. So what about AMPAS1 neurons, right? We kept saying like they have to be important for motor control, right? Is that the case? So similar layout to what I showed you before, this AMPAS1 neurons, we're shining light onto the cell body, and we're measuring movement here. And see, when we shine light here, there's a dip in the movement, okay? And this is not reproduced when only EYFP is expressed, okay? So when we shine light in the striatum, activating the terminals of AMPAS1 neurons, we can see a very weak suppression of movement. It is not entirely surprising just because the volume of the striatum is so big, and we can only shine light into very restricted area within the striatum using a fiber. So, but we nevertheless saw a consistent decrease in movement. Okay. So can we inhibit AMPAS1 neurons and see something opposite? And the answer is yes. Again, this the inhibitory opsin that we introduced into AMPAS1 neurons, and by shining light onto AMPAS1 neurons, we can actually see an increase in movement. So this is not just a weird gain of function experiment that gives us something that may not be physiological, because we can actually see it by manipulating the neurons in the other way. So remember, I told you earlier that FOXP2 neurons is a subset of AMPAS1 neurons. It turns out when we activate those neurons, we can actually see even a more remarkable suppression of motor output than when we suppress only all AMPAS1 neurons. Okay, this is the perfect control. Again, it's when only EYFP is expressed. So overall, the behavioral assays is consistent with anatomical data, right? Saying that PV and AMPAS1 neurons actually have distinct function, which match with the distinct axonal projections of these neurons. So let's get back to the early model that I introduced to you. in the earlier part of the talk. This is again the model from Alban. He basically again said the GPE is a black box, but now we know that's not quite true, right? There are two types of cells, and we know that it's not homogeneous, right? And it's not just doing a relay of information from the striatum to the ST, it's actually going the opposite direction as well. So we have added new information to the model. So let's summarize what I told you today. Today I told you that we can identify cells in the GPE. They're basically two cell classes, right? They can be identified based on the expression of PV or MPAS1. And we have used a combination of metrics to identify them and to tell them they are indeed apart from each other. They have different functions. Again, PV neurons are motor promoting, and MPAS1 neurons are motor suppressing. And we also highlighted a few changes in the circuit connectivity or output that can be potentially important in explaining why patients have hypokinetic symptoms. We have also mentioned a few things about potential subtypes within the GPE, and hopefully we can map those neurons onto different maybe fine movements or different functions, other than just controlling motor. So there's an enormous amount of work produced by lots of people from the lab, both current and past members. I'm not going to tell you individually what they did. I will want to highlight a few things. Vivian was my first PhD student. She was the major driving force in terms of making the mouse, making the antibodies, and describing the two neuron types within the GPE. And this ongoing action from the lab, from the people here in the middle, trying to tell subtypes of neurons apart from each other. The behavioral assays were done mostly by Charlene Quay, postdoc in my lab, and Erin Pamukchu, which is about to graduate her PhD. Kelly and Harry spent a lot of effort in describing the connectivity between the GPE and the striatum. And of course, we have help from lots of people. Nick was involved in making the mouse. Bianca Lim has sent us a lot of viruses. And I didn't get a chance to talk about the input mapping data, but we got help from Young-Soo Kim. Raj was helping us with identifying new markers for different neuron types. And Taylor Lerner is in the department. She helped us with the behavioral assays. And also got the help from Levenger for some electrophysiological analysis, and EM data was contributed from Charles Smith-Sewell. And we're doing some modeling with Josh Plotkin, and some new exciting collaboration with Ellen Smith and studying the cortical GPE loop. And finally, these are the funding sources. And before I end the talk, I'll just leave the circuit model up. It's a little more interesting to look at in case you want to ask me circuit questions. And I'll just stop here. Thank you for listening. I'll be happy to take any questions.
Hi, that was a very interesting talk.
Thank you.
You talked a lot about how you were able to separate the GPE into two distinct populations.
Right.
But something that struck me was at the beginning when you were talking about the deep brain stimulation, just sticking a wire down in the brain and non-specifically just activating whatever is in the neighborhood also works. So how do you explain these two distinct populations that seem to have opposite mechanisms, and yet just stimulating everybody together seems to work?
That's a good question. So let me bring up that slide. So I may have not explained it well enough. Oops. So this electrode is actually targeted the STN, right? So essentially, what can I do to So it's actually targeted the STN. So you can imagine that could be more effective, or should be more effective, in recruiting the axons coming from the PV neurons, right? But not so much the MPAS1. Although the MPAS1 neurons do send axons right above the STN. So it depends on the targeting. But we know that to get very effective DBS, this has to be like skewering the olive really, really well. It's not just like in the general area. But we think it's more complicated than that. So probably it's through stimulating PV neurons, but also there are a lot of stuff passing through the STN. In addition to PV axons, we know cortical axons actually go through the STN as well. Cortex innervates the STN heavily, right? But people now know that if you put this stimulating electrode into the GPE, it works as well, right? Maybe just as effective. There's still a lot of data that are coming out. We don't fully know yet, but probably why we want to study the cortex GPE interaction, because it's probably because of the deep brain stimulation data showing that stimulating the GPE can be just as effective. So potentially, what DBS is doing is recruiting cortical neurons rather than recruiting GPE, but it's likely a combination of all the above.
Back here, Savio.
Yes.
So fantastic talk. I think this combination of these molecular screening with electrophysiology, this like 22nd century dissection neuroanatomy, it's beautiful.
Thank you.
So one question I had is, now that you have these cell-specific markers and promoters, particularly in these models, whether it's the Parkinson model, let's say another mouse model of neurodegeneration,
Right.
have you begun then looking at selective expression of things in these cell types that might rescue the wild type phenotype?
Right, that is a great question. That is really the hope, right? So I don't think we're quite there yet. So let me bring you to. This. So why are we spending so much time doing this kind of combinatorial analysis? Because people have already done single-cell RNA-seq, right? So maybe we don't need new markers, but the answer is yes, we want to have a more effective way to screen and identify cells. And the fact that just because we have subtype of neurons, by using only PV mouse line or MPAS1 mouse line cannot give us enough specificity, right? So we're digging through the RNA-seq data, and in the meantime, we are trying to find useful markers and also useful mouse line. We're going through that process now, so maybe in a few years, I can come back and say like, yep, we can effectively manipulate different cell types. We can express different things in different cell types and seeing restoration or seeing whatever. That's the hope.
Any more questions?
With your DREAD study, I was curious because you showed basically some gross motor inactivity, right?
Mm-hmm.
And I wondered because as you started out, you were showing the PD patient and how once they were treated with the deep brain stimulation, they were able to do some tracking and motor saccades that were where he could point to the finger. So I guess my question is, have you extended that into looking at more goal-oriented behavior rather than just gross motor activity?
We've been thinking about that. We're not behaviorists. What we're good at is actually doing recording, electrophysiological recording. So this is kind of like our first pass, and then the optogenetic analysis is kind of like what we're doing now, but we should be doing something even finer than that, at least in terms of like measuring motor output. Can we measure finer output on a smaller time scale? And in terms of goal-directed behavior, we've been thinking about that a lot, especially because of our interest in studying the cortex. Where it seems like there's a reward circuit type stuff that's kind of like built in there, or learning circuit that's built in there. Except that I think it's a little difficult or tricky to dissociate learning or goal-directed behavior from reward. So that's kind of like, we don't quite know what to do yet. We've been thinking about that a lot. So likely that is the direction, but we want to recruit collaborators.
Because you also set it up and said, I'm not going to be talking a lot about dopamine today, because that's going to be another composite as you broke down different subtypes within cellular subtypes within the external globus pallidus. So, was there a reason behind that? Was it just here's my niche and here's what I'm going to add to the literature? Or
no, we didn't really try to leave out dopamine.
Yeah.
Haven't really thought about this question so much. We had screened for dopamine receptor expression like years ago, and it was just kind of a mess before we knew more about cell type. And also, that was also done probably with single cell PCR, so that wasn't quite clean. But if you look at dopamine innovation, it's not a ton, right? So that's probably why we don't want to look at dopamine function per se yet. However, we know that while there's not a ton of dopamine innovation, there's also no data expression. That means once dopamine is released or dumped, it's going to be lingering. So they can have profound functional consequences. So it's just something that we just haven't gone to yet.
Okay.
Yeah.
Thank you.
Thank you.
Oh, wow, thank you. Very interesting talk, and it's kind of a follow-up to
Thank
both Chris and Marcos's question. In thinking about, NPAS, right, transcription factor, so you've heard a lot about sleep and circadian rhythms today. So PAS, right, per aren't sim, so per is period, right? So it's basically helix loop helix. NPAS2 can emulate clock in the absence of clock to rescue some of the circadian stuff. So where I'm going with this is initially you show a lot of physiological data about firing rates. So one of the great mysteries right now, trying to understand the biology of these transcription factors, particularly in the circadian system, is how does that translate to firing rate? And so you very elegantly show there's something about these NPAS1 neurons, and you've also done a lot of the discovery-based molecular strategies to identify unique signatures within these cells. Can you identify anything like channels or, so what is it about NPAS1 to regulate the circuit and regulate firing rate?
Right. Great question. It's something that I should know, but I don't actually know. It's kind of like what we're using PV for. We're mostly using that as a molecular marker rather than trying to understand the function. So at least at the beginning, we have made a cross of our NPAS1 reporter mouse with the NPAS1 knockout. We didn't see any profound changes. That's kind of like tell us, okay, let's not pursue that so much. But if you go look at the literature or the papers from John Rubinstein, I think he described it quite elegantly or quite convincingly that NPAS1 along with NPAS3, which is kind of like doing something what NPAS1 is doing. They have right. Steve McKnight was probably the earliest, but since then, I think John Rubinstein continued to make the contributions. But you're right, Steve McKnight is an important figure in this story. I think maybe you know the literature more than I do in that sense. They basically said that those transcription factors are important in regulating cell fate and also the maintenance of the fate, but there hasn't been a ton of electrophysiological analysis done from the mutant mice. I think probably because of the fact that John's lab or Steve's lab are not they focus mostly on the molecular aspect or the developmental aspect, not so much the electrophysiological aspect. Well, although they do collaborate, for example, with Scott Barron. But so we have talked about potentially doing so, we just never gotten a chance yet. So hopefully we'll know. But I think maybe MPAS-2 and MPAS-4 are more dynamically involved in regulating activity-dependent changes, but I have not seen a lot of similar type of plasticity work describing how MPAS-1 and 3 are involved. So.
Any more questions? So then join me to thank Savio for a wonderful lecture.
Oh, yeah. Thank you.